in central dogma, this DNA what it gives? This DNA is a double-stranded helix to form up only mRNA. mRNA is a single-stranded helix to synthesize only protein. To synthesize only protein. This process transcription. So DNA converted to mRNA. That process is also called as transcriptions. Transcriptions. These mRNA to convert to form of only proteins. This process is also called as translations. This process is also called as translations. These DNA, RNA is a genetic material in presence of only all living organisms, plants and animals. Plants and animals. These formation of RNA and mRNA. These formation of RNA or production of RNA bases are classified to in viruses. How to form up only mRNA? The mRNA form up only from the process only transcription. From the process only transcription. DNA convert to mRNA. The production of mRNA. Production of mRNA. The first types of classifications. The second is nucleic acids. The nucleic acid, DNA is a nucleic acids, RNA is a nucleic acids. These type of nucleic acids found in only all living organisms, plants and animals. But the virus possesses only either DNA or RNA. The virus possesses only either DNA or RNA, but both never. But both never are present only in viruses. The third types of classification, the host species. Some other virus infected in plants, some other virus infected in animals, some other virus infected in fungi, some other virus infected in cyanobacteria. That is a blue-green algae. Cyanobacteria, blue-green. These are only found in only host-specific organisms. These are based on classification. There are three categories. The first one is on the basis of RNA production. The second category is on the basis of nucleic acid or genome. Third category is on the basis of host species. These type of classifications. This type of classification is given by. This type of classification are given by David. This type of classification is given by David Baltimore's in the year of 1971. In the classification on the based on mechanism of RNA productions. Based on mechanism of RNA productions. Based on mechanism of RNA productions, so given by David Baltimore. The first point is based on the classifications, based on mechanism of RNA productions. Mechanism of RNA production. The mechanism of RNA productions, DNA converted into RNA. The RNA converted into protein synthesis. RNA converted into protein synthesis. Protein synthesis. This DNA converted to RNA. This process is also called as transcription. Transcription. The RNA converted to protein synthesis. That process also called as translations. Translation. These again, these RNA reconverted to DNA. These RNA reconverted to DNA. That is called a reverse transcriptions. Reverse transcriptions. Reverse transcription. The first point is based on the classifications, based on mechanism of RNA productions. Mechanism of RNA productions. These RNA productions. How is form of RNA production? The DNA converted to RNA. The RNA converted to protein synthesis. The DNA converted to RNA. That process also called as transcription. The RNA converted to protein synthesis. That is called a translation. Again, these RNA reconverted to form of only DNA. It's a double strand of helix of DNA structure. The single strand of helix of only these RNA reconverted to the process also called as reverse transcription. Reverse transcription. The next one is nature of genomes. The nature of genomes. Because
audio மட்டும் போயிட்டு வீடியோ வந்து லிங்க் கட் ஆயிடுச்சு கனெக்ட் பண்ணி
this the second point is nature of genomes. The nature of genomes, either single strand of DNA or double strand of DNA. So genome is a haploid set of chromosomal structures. The haploid set of chromosomal structures that is called genomes. These single standard DNA or single standard RNA structures. The genome like that, single standard DNA or single standard RNA. Either these DNA, double standard DNA structures or double standard RNA structures. The nature of genomes are found in only virus, either a single standard or double standard RNA or DNA. RNA or DNA. The single strand of DNA or double strand of DNA, a single strand of RNA or double strand of RNA structures are found in only in virus. Next, the use of reverse transcriptation. By these process used in how is form of this only RNA? By this process of only use of reverse transcription. Transcriptions. Reverse transcription. So what is reverse transcriptions? These DNA molecules, the double strand of DNA molecules, are converted to form of only mRNA groups. mRNA refers to messenger RNA. Messenger RNA is a single strand of RNA. This single strand of RNA converted to form of only protein synthesis. It's a protein molecule. So China protein. These mRNA reconverted to single strand of RNA reconverted to DNA structure. Double standard either the single strand of DNA or protein. This process also called as reverse transcriptions. Reverse transcription. These 3 points are involved the basis of only RNA production. The next one is these are formed. These reverse transcriptions are formed in single strand of RNA. Single strand of RNA may be produced in 2 types of RNA structures. The first one is the positive sense of RNA. Positive sense of RNA or negative sense of RNA. The positive sense of RNA or negative sense of RNA. These negative sense of RNA also called as antisense. Antisense. So what do you want antisense? These positive sense of RNA These type of in presence of only positive sense of RNA, synthesis of only these positive sense of RNA, the synthesis of only protein molecules. Protein molecules. These type of RNA. This type of RNA. This type of positive sense of RNA. Positive sense of RNA. DNA is converted to mRNA. mRNA is mRNA converted to form of only synthesis of protein. Synthesis of protein. The protein synthesized from only mRNA. In between form of only mRNA is only positive sense of RNA. Positive sense of single strand of RNA. But these negative sense of RNA, that's the antisense of RNA, these RNA molecules, these mRNA molecules does not produce protein synthesis. Does not produce in proteins. Does not produce in protein. But which type of RNA does not synthesize in antisense RNA? Antisense of RNA. But positive sense of RNA synthesis of only proteins. But positive sense of RNA synthesis of protein. The viruses are classified into 7 categories. The viruses are classified into 7 categories. Who's the first 7 categories of viruses are classified in year of 1971? David Battimore. 7 types of classifications. So what are the 7 types of classifications? The 7 types of classifications. The 7 types of classification given by the tables. You can see the textbook. The given by David Battimore in the 1971. These 7 types of classifications. 7 types of classifications. Classification was first introduced in these 7 types of classifications. David Baltimore in the year of 1971. Here given this chart in textbook, the first class 1. The class 1 viruses with DNA, double strand of DNA. Viruses with a double strand of DNA. Already you know in this classification based on double strand of DNA, nucleic acids, single strand of DNA nucleic acids, double strand of RNA nucleic acids, genome, the genome classification based on. These are genome classification. And single strand of RNA, genome, then positive sense of RNA, single strand of RNA. Then negative sense, single strand of RNA. Negative sense, single strand of RNA. Negative sense of single strand of RNA. The next one is double stranded DNA with reverse transcription. These are the 7 categories. These are viral genomes. What are the viral genomes? The double strand of DNA is a viral genome. Single strand of DNA is a viral genome. Double strand of RNA is a viral genome. Single strand of RNA is a viral genome. Is a positive sense of RNA is a viral genome, and negative sense of single strand of viral genome, and double strand with the reverse transcription. These are 7 categories are classified given by David Battimore in the year of 1971. The first class 1, virus with the double strand of DNA. The first class 1, the virus with the double strand of DNA. These are double strand of DNA. Double strand of DNA. 3 dash helix opposite the 5 dash helix. The 3 dash helix opposite the only 5 dash helix. 5 dash opposite the only 3 dash helix. These DNAs convert to form of only mRNA. This DNA is convert to form only mRNA is a single strand. These are converted. We convert to form only the synthesis of protein. The synthesis of protein. The type of virus called viruses with the double strand of DNA. The virus with the double strand of DNA. With this type of virus are examined in adenoviruses. Adenoviruses. The second class, class 2. In class 2, viruses with the positive sense of single strand of DNA. The positive sense of single strand of DNA. See, positive sense of single strand of DNA is positive sense of DNA or RNA, the of only protein also. The same type of only in double strand of DNA, but these DNA to produce only a single strand of only DNA molecules. Single strand of only DNA molecules. These DNA convert to form only mRNA. These mRNA convert to are synthesized in protein. It's a positive sense of the positive sense of DNA or positive sense of RNA, synthesis of only protein. Synthesis of only protein. The type of virus that is called a parvoviruses. Parvovirus. The parvovirus possesses only positive sense of single strand of DNA. So very very important one more question. Where it is presence of only single strand of DNA, these single strand of DNA found in only parvoviruses. Parvoviruses. The third class, class 3. In class 3, viruses with the double strand of RNA. The viruses with the double strand of RNA is example for only reoviruses. Viruses with the double strand of RNA, example in reoviruses. These all are produced protein species. The adenovirus. Class 1 is of protein. The class 2 also is of protein. The class 3 also is of only protein. Because these RNA transcription and translation process are involved. Transcription. DNA convert to RNA. Transcription process. RNA convert to protein. It's a translation process. These both processes are involved only in this type of virus. So synthesis of only protein. So synthesis of only protein. That type of virus also in reovirus. What's the example for this only? Is double standard of RNA. Double standard of RNA reovirus. The class 4. Next class 4. The viruses with the positive sense of single strand of RNA. Positive sense of single strand of RNA. These positive sense of single strand of RNA also, because the presence of only positive sense, the sense of only protein. DNA convert to RNA, RNA convert to protein. The protein synthesized in viruses with the positive sense of single strand of RNA. Examples: togoviruses. The next one is the class 5 viruses with the negative sense of single strand of RNA. The negative sense of single strand of RNA also called as antisense RNA. Antisense RNA, there is no sense of protein. But this type of genome is active. But the single strand of genome, this type of genome is active. But there is no synthesis of protein. But no synthesis of proteins. Synthesis of protein. But genome is active. The virus possesses a single strand of RNA, but this viral genome is active, but there is no synthesis of protein. Examples: rabidovirus. That's class 6. Class 6 viruses with positive sense of single strand of RNA. Reverse transcriptase. With that replicate with the DNA. What's a replicate? Say replicate means replication of the genome. Like the same types of genome they produced. Same types of genomes are produced. For example, this type of DNA structures. Is a single strand of DNA again produced in double strand structures. This single strand of DNA produced again double helix structures. This single strand of DNA again produced double strand. These single strand of DNA are produced again double strand of structures. These are only DNA structures. These are two DNA structures are found in DNA structures. Again, 4 DNA structures. 4 DNA structures. 4 DNA structures are produced. 1, 2, 4, 4 again to form only. These are only replications. The replication of own genomes. Replication of own genome. Let's say replicate with the DNA. Replicate with the DNA. Intermediate in the life cycle. Intermediate with the life cycle. For example, this type of class 6 virus with the positive sense of single strand of RNA. Positive with a single strand of RNA. For example, these are single strand of RNA. There is mRNA. mRNA is a single strand of RNA. These single strand of RNA to form up only reverse transcriptions. Reverse transcription. The sense of only DNA. The DNA is a old. This already you know DNA structures. These DNA structures again replicate. Again replicate with their own genomes. Your genome these to convert to form only mRNA. Then these mRNA is only proteins because it's a positive sense. Because it's present only positive sense of single strand of RNA. That reverse transcription process is replicate with the DNA, intermediate with the life cycle. Examples: retrovirus. The class 7 virus with the double strand of DNA. Viruses with a double strand of DNA, immediate to form only reverse transcription or replicate with the RNA. Replicate with the RNA, intermediate the life cycle. Intermediate with the life cycle. These are 7 types of classifications given by David Baltimore's in the 1971. The next one is viral genomes. What is a viral genome? Is DNA strand and RNA strand all are viral genomes. The viral genome possesses nucleic acids. That is either DNA nucleic acids or RNA nucleic acids. Next one is the viral genomes. Viral genomes. Viral genome. Each virus possesses only one type of nucleic acid. Each virus possesses only one type of nucleic acids. Nucleic acids, either DNA or RNA, but both never. So each virus possesses only one type of nucleic acids. Only one type of nucleic. What type of nucleic acids? Either DNA helix or RNA helix. For example, these are, tobacco mosaic virus. This tobacco mosaic virus, single strand of RNA. Only found in only RNA. These living organisms possess. Covered by the protein pools. It's only possess. This type of RNA viruses possess only RNA, either DNA. The nucleic acids may be linear or circular. The nucleic acids, this type of nucleic acids, either linear or circular. So DNA strand is in human being. Maximum prokaryotic organism DNA structures. Maximum prokaryotic organism DNA structure circular. In prokaryotic organism, in examples in bacteria, in circular. circular. In lineage, some other viruses possess only lineage. Lineage stages. Each virus possesses only one type of virus. It's a nucleic acid, say the DNA or RNA. Nucleic acid contains linear or circular. Some virus DNA possess. Some virus DNA possess the type of virus also called deoxyviruses. What do you mean deoxyviruses? Some virus having only double strand of DNA or single strand of DNA. Either double strand of DNA, either single strand of DNA. Some virus having that type of virus also called as deoxyvirus. So DNA refers already told you. Deoxyribonucleic acids. Deoxyribonucleic acids. So deoxyvirus. Deoxyvirus. Some virus possess RNA. Some virus possess RNA, either double strand of RNA or either single strand of RNA. Double strand of RNA, either single strand of RNA. That type of virus also called as riboviruses. Riboviruses. Ribovirus. What about deoxyvirus? Some virus having only DNA structures. DNA strands. That's the DNA double strand. Some virus having only RNA strands. That type of virus also called as ribovirus. The viral genome contains either only nucleic acids, either DNA nucleic acids or RNA nucleic acids. The nucleic acids are composed of chemical substances. The nucleic acids, what is the nucleic acid? The nucleic acid is a composed of only chemical structure. Nucleic acid is a chemical structures. Chemical substances present. What types of chemical substances are present? It's some types of chemical substance. It's found in only nucleic acid is found in only nucleotide and nucleoside. Nucleoside. What's a nucleoside? The nucleoside is composed of only 3 chemical compounds. So what are 3 chemical compounds are present? The first one is nitrogenous base. First one is nitrogenous base, sugar substances, pentose sugar and phosphate. There are 3 chemical compounds are present in nucleotides. The first one is nitrogenous base, second one is pentose sugar, and third one is phosphate. But these nucleotides presents nucleoside and phosphate. Nucleoside plus phosphate. The nitrogenous base are only pyrimic bases and pyrimidine bases. Pyrimic bases and pyrimidine bases. These are nucleoside. These nucleoside contains only 2 chemical compounds. One is pentose sugar. These are only nitrogenous base plus pentose sugars. Pentose sugar does not having only phosphate. Phosphate absent only nucleosides. The nucleic acid contains some other chemical compounds are present. What types of chemical compounds are present? Nucleotide and a nucleus. That is a nitrogenous base, pentose sugar, and phosphate. That's this type of only viral genome. Next, majority of animal virus. The majority of animal virus possess only RNA. The majority of animal virus possess in majority of animals and bacteria possess DNA cells. In majority of animals and bacteria cells contains only DNA is a genetic material. DNA is a genetic material. Except in HIV, human HIV, human immunodeficiency virus, HIV contains only RNA is a genetic material. In human, it's a human immunodeficiency virus. Is immunodeficiency virus contains only animal virus. These animals virus presence of only RNA is a genetic material. But the majority of animals and bacteria cell contains only DNA is a genetic material. DNA is a genetic material. But except in HIV virus possesses only RNA virus. Possesses only RNA virus. The plant viruses. In plant viruses contains only RNA. The plant virus contains only RNA, but in cauliflower mosaic virus, in plant, but a cauliflower mosaic virus. Mosaic virus contain only DNA. It's a cauliflower mosaic virus contains only DNA. In majority of animals and bacteria cells contains only DNA is a genetic material, but except in HIV virus contains only RNA is a genetic material. The plant virus, majority of plant virus contains only RNA, but except in cauliflower mosaic virus contains only DNA cells. Cauliflower mosaic virus that is called DNA. So nucleic acids may be single stranded or double stranded. Nucleic acids may be single stranded or double stranded. Single stranded or double stranded. There is a single stranded or double stranded. The DNA is a double stranded helix, RNA is a single stranded helix. But in Wund virus, a single unit. Majority of virus, only a single unit of virus. Nucleotide genome. In nucleotide genome, that is called a single unit. But in Wund tumor virus, contains only segmented viruses. Segmented in genomes. For example, so most of the virus covered by the protein codes. These protein codes are nucleic acids only in segmented in wound tumor virus. But majority of virus is covered by the protein codes. It's a single unit genome like that. The majority of viral genomes, single units of viral genomes, but these Wund tumor virus contains only the segmented genomes are found. These are only viral genome pairs. Yes, on the basis of natural nucleic acids of 4 classes. On the basis of nucleic acids based on the 4 classes. And the nucleic acids based on the 4 classes. So what are the 4 classes based? The first one is on the basis of nucleic acids. On the basis of nucleic acids, there are the 4 classes. There are the 4 classes. So what are the 4 classes based on only nucleic acids? The first one is viruses with a single strand of DNA. Virus with a single strand of DNA. Then virus with a double strand of DNA. The viruses with a single strand of RNA. Then virus with a double strand of RNA. Double strand of RNA. So viral genome contains only the DNA and RNA. In the DNA and RNA, it's based of nucleic acids. Based of nucleic acids, there are four classes. The first one is virus with a single strand of DNA. The viruses with a double strand of DNA. The viruses with a single strand of RNA. The virus with a single strand of double strand of RNA. Double strand of RNA. These are possess only positive cells or either negative cells of RNA. Negative cells of RNA. These virus single strand of RNA examples. For these examples, paravirus, parvovirus. Example for double strand of DNA, bacteriophage. Bacteriophage. The viruses with a single strand of RNA examples, tobacco mosaic virus. Tobacco mosaic virus. The viruses with the double strand of DNA example for this only one tumor virus. Tumor virus. These are example for this only. The parvovirus is example for a single strand of DNA. So bacteriophage is example in the double strand of DNA structures. The tobacco mosaic virus example in only in single strand of RNA. The one tumor virus example in only double strand of RNA. These are basis of nucleic acids, four classes. Again, on the basis of host species. The third classifications on the basis of host species. The next one is the third classification methods on the basis of host species, host specific organism. The virus infected in animals. Some virus infected in bacteria. Some virus infected in plants. Some virus infected in algae. Some virus infected in only in blue-green algae. These are only on the basis of host-specific organism. Host-specific organism. For example, this virus, Jupiter. This type of virus, bacteriophage. And third one is bacteriophage. The third one is phytophage. Then fourth one is phytophage. Then fifth one is mycophage. Then sixth one is cyanophage. Cyanophage. These type of classifications are based on only host species. Host species. Some virus infected in animals. Infect in animals. That type of virus is also called as Jupiter. Phage refers to virus. Phage refers to virus. Zoo refers to animals. Zoo refers to animals. Some virus infected in animals, the type of virus also called as zoophage. Some virus infected in bacteria. Infected in bacteria, the type of phage is called as bacteriophage. Infected in bacteria. That is called as bacteriophage. Some virus infected in plants. That type of virus also called as phytophage. Some virus infected in algae. Infected algae. That type of virus also called as phytophage. Some virus infected fungi. Fungi. That type of virus also called as mycophage. So some virus infected blue-green algae. Blue-green algae, that is called a cyanophage. Cyanophage, there are these six classifications on the basis of only host species specific organism. The June page, second one is vector page, third one is spider page, fourth one is micro page, the fifth one is micro page, and sixth one is silent page. These are some virus infected some other organism. Based on the conditions on the basis of only host species. These are only a classification of viruses. Classification of viruses. The next one is tobacco mosaic virus. Tobacco mosaic virus. It is a rod-shaped virus. There are already known shape of the virus. There are three types of shapes in virus. The first one is helical shape, symmetry, cuboid shape, symmetry, and rod shape, symmetry. Rod shape, symmetry in tobacco mosaic virus. Tobacco mosaic virus. This tobacco mosaic virus in rod shape. Tobacco mosaic virus. It discovered by who's discovered by the tobacco mosaic virus? Dimitri Ivanitsky. Dimitri Ivanitsky. Ivanitsky, year of 1892. 1892. Where is discovered this from? Tobacco plant. From tobacco plant. That is called a tobacco mosaic. These virus patterns are in the mosaic pattern. Mosaic patterns. The virus infected symptoms appearing only mosaic patterns on the leaves. On the leaf, it's a leaf-like structures. These virus are in the decolorations to form up only arranged only in yellowy colors. Decolorizations, the arranged only in mosaic patterns. Or veins or margins of leaves. Mosaic patterns, veins or margins of leaves. That is also called as tobacco mosaic virus. Who is the first discovered the tobacco mosaic virus? Dimitri Ivanitsky. When will be discovered by? There of only 1892. 1892. The second is the virus infect the healthy plant. The virus infect in healthy plant. Healthy plant through the vectors. Some vectors carry the virus. Some vectors are carrying out the virus infected by this only tobacco mosaic patterns are arranged. The first one is aphids and lacunes. Aphids and the aphids is some type of only sap sucking insect. The aphids that is called a sap sucking insect. It's a kind of sap sucking insect. From this only on the leaf, decolorization. The protein sucking and decolorization formed in mosaic pattern. That type of process in mosaic tobacco mosaic virus. Second, the symptoms may be appearing only decolorizations. The symptoms. What are the symptoms is appearing in the tobacco mosaic virus? Symptoms. One is decolorizations. Decoloration on leaf. Which mosaic symptoms? Which mosaic symptoms? Mosaic symptoms. So downwards, the symptoms appearing downwards. The symptoms appearing only downwards, curly and distortion of young epitheliums. Distortions of young leaves. Young leaves. These are only tobacco mosaic virus characters. Who is the first in the Dimitri Ivanitsky in 1892 first discovered by this tobacco mosaic virus from this tobacco plant. We infected these some healthy plants through these vectors, such as vector that is called aphids and acutes. Symptoms appearing. It may be symptoms appearing: decolorization, downwards curling of leaves, distraction of young leaves, distraction of young leaves. The plant becomes stunted. Plant becomes stunted. The plant become stunted. The plant becomes stunted and yield is affected. Yield is affected. These are symptoms appearing only in tobacco mosaic virus. Tobacco mosaic virus. It may be to find out. That's all about this topic. This topic, please. Just recall for this topic. The first is the MF virus. The second one is classification of virus. Classification of virus.
Students, unmute your phone and answer one more question.
The first suppose the discovered by this only classification based on the RNA virus.
Anyone can answer this question?
Can be the answer first. Who's the discovered by the mechanism of RNA production based on the classifications?
Akhilan, Chegwara, Maharajan, Ashwini.
David Baltimore.
Tell again 1 second.
David Baltimore.
David Baltimore. So when will you find out this?
Baltimore.
Very good. The second question is, what is the reverse transcription? Anybody? Reverse transcriptions. I understand this topic. The reverse. In central dogma, the DNA converted to RNA, RNA converted to protein synthesis. So DNA is converted to RNA, that is called a transcription. The RNA is converted to protein, that is called a translation. What is reverse transcription?
Anyone can give answer for reverse transcription?
Sir, the use of reverse transcriptors, single stranded RNA may be positive sense or negative sense is antisense.
Nitish, what is loop? Only